So our approach for tackling this challenge is by using CRISPR. But what we really cared about and what we drew inspiration from is that CRISPR is actually an adaptive immune system for bacteria. The way that it works is that when a bacteriophage or a bacterial virus infects a bacteria, specific proteins in the bacteria will take snippets of the bacteriophage genome and incorporate it into its own genome as a guide RNA. And this is important because the next time the bacteria encounters a virus it has already seen, it will use these guide RNAs, like molecular zip codes, that direct specific CRISPR-associated proteins to these invading viruses and destroy them, thus protecting the bacteria from infection. So our thinking was, instead of interfering with the viral proteins, what if we choose to instead interfere with and destroy the viral RNA? This would allow us to attack the virus in a completely different manner and give us a number of opportunities to do so. So overall, our goal, of course, was to test whether or not we could use CRISPR to eliminate SARS-CoV-2 in live cells. However, Stanford does not yet have a facility with a high enough biosafety level to handle live SARS-CoV-2. So instead, we synthesized and used snippets of genes from SARS-CoV-2 that were highly conserved for this virus, SARS, and MERS viruses as a proxy. For one, all we really need to know about the virus is the sequence of its genome, which is incredibly fast and cheap to find nowadays. Developing effective vaccines and drugs, on the other hand, takes a lot more manual labor and time to screen through and find effective targets. Additionally, there may be a lower likelihood that highly conserved regions of the viral genome mutate, as they may be essential for the virus's function. So by targeting these regions, there may be a lower chance that the virus mutates and escapes therapy, as was shown earlier about influenza. We may also be able to target many different strains of virus, like SARS-CoV-1 and MERS, that all share this conserved sequence. Third, it's easily multiplexable, meaning that we can introduce a number of different guide RNAs simultaneously. This has the advantage in that we can target multiple regions of one virus, further reduce the probability of mutational escape, and we can design a small set of guide RNAs that can target all sequenced coronaviruses or influenza strains. So for example, we found that with just 6 guide RNAs, we could potentially target 91% of all the known coronaviruses, and that with only 22 guide RNAs, we could target potentially all of them. Lastly, we can combine our approach with the traditional therapies for potentially an even greater patient response. So having a multi-pronged approach using vaccines, CRISPR, and small molecule drugs to interfere with the virus. I really think that it could, because as I mentioned, we only really need the sequence of the viral genome. And once we have the viral genome and can find potential targetable spots, it's very easy to just print out guide RNAs and test those and find those that are very capable of reducing the amount of viral load. And we can really extend this to any sort of RNA virus that we want. So we've so far looked at SARS-CoV-2 and influenza, but you can only imagine that you can send that to other RNA viruses. I think hepatitis, I think Ebola, Ebola as well is an RNA virus. So really, this approach could be extended to really any RNA virus that you're interested in. Ultimately, there are a number of questions that we need to answer and a number of tests that we need to do in larger organisms and in humans before we can get this by and approved by the FDA and used in patients. So unfortunately, it won't be ready for this pandemic, but our hope is that for future pandemics, we would be able to use this. Yeah, that's pretty much all of what we're looking into. So I mentioned by making the protein from bacteria or other sources, or making adeno-associated viruses or RNAs. So the eventual end goal would be pretty much to try and use like an inhaler-like system, where you just take the drug and the person just takes a couple of puffs of that, and it gets then delivered into the lungs where it can exert its effect on the viruses.